article on the analysis of COVID-19 RNA Seq data sets, trying to find differentially expressed genes using the Seq2. So this is a huge course on its own, but we'll try our best just to get some kind of knowledge in this section. Right. Okay. So, all right. So we know that the canonical ideology of biology, we know that DNA it transcribed into pre-mRNA, and then the pre-mRNA goes through splicing and then capping and tailing, and then we get the matured mRNA, and then the matured mRNA is translated into proteins, right? So someone will ask, what is RNA-seq, right? So for RNA-seq, we just consider the matured mRNA, right, from the genes in consider maybe tissue or single cells, and we try to detect how these genes are transcribed, right, given various groups or various groups, or let's say various sample size, or we can even look at it from different time frames, right? But others do also look at the protein profiles of these genes to understand how these genes are transcribed, and then how these genes can help in other biological processes. So now we go through the RNA-seq workflow. So the first part is the biological samples, and then where the samples are harvested, and then the RNAs are isolated, and then the matured mRNAs are selected by their poly-A tail to be used in sequencing, right? Now, after sequencing, these generate millions of short sequences that are called reads, right? So we have the sequencing reads, and then the sequencing reads are output as a FASTQ file, right? And then after getting the FASTQ file, now there's an option that is looking at your data set, your FASTQ file, to see whether there's no contamination from cDNA, right? And then looking if your reads are very good for you to use for the analysis. Now, the next part is to map these reads to the various genomes, because these reads are sequenced. So we need to know which part of these genomes these reads belongs to. So we map these reads to the various genomes. And now the last part is to count these reads. So the number of reads that are mapped to the various genome, it's counts, and then they are associated with the gene at that particular site. And then we can either save it in a text file or any kind of file, which is called a count matrix. But for this section, we are going to consider or concentrate mostly on identify differential expressed genes, because all these workflow or all these processes can be done in command line tools, and then it takes a lot to go through all these processes. So we're just going to consider now, after getting your RNA Seq data file in a form of a text file, right? How are you going to identify genes that are upward expressed or downward expressed in two various groups, right? So for this session, the data set that we're going to use is taken from this paper called Differential Gene Expression of SARS-CoV-2 Infection of Primary Human Lungs Epithelium for COVID-19, right? So, and this is the G code. So when you click on this link, it will send you to where the data set is. And since I've already done that, downloaded it, and then it's part of the GitHub, so you have everything there. So from this paper, what they did was they were trying to look at various genes that are being affected by this COVID-19, and this will help them in generating drugs or trying to, come up with drugs for this COVID-19. So they have 5 mock sample size, that is a vehicle data set, and then 5 samples from infected COVID-19. So the total number of samples are 10, right? So somebody who asked what is differential gene expression? And then, so this is a technique to determine whether genes are expressed at significantly levels between two or more sample groups, right? And then the goal is for each gene, whether the difference in expression between the groups are significant, right? Given the amount of variation within groups or between biological replicates. So from here, we will just move to the handout session, and then, all right. So can you please see my screen?
Yes, we all can.
Okay. All right. So normally platforms, or there are several softwares that are used in differential gene expression, but one of the one of the common one is use R, and then there's this library that is called RNA Seq, and then this helps because they've already written everything in, and then it helps us to know or to reveal these genes that are differentially expressed in one group, and then let's say upward expressed in one group and then downward expressed in the other group. So first, let's look at, I want to know how many people knows how to use R or do have a hands-on experience in using R.
You can reply by raising your hands. It's on the reaction emoji icon. Well, we have one person saying no, sir.
Okay. And then I hope we all had these resources before today. So because if you want to follow, there are some packages and then installments that you need to do, which was provided in the documents that I sent. So
we did. We sent out the email for all the applicants who registered. So it's been taken care of.
Okay, thank you. So then let's move on. So this is, let me first show you how the RNA Seq data set looks like. So this is how the RNA Seq data set looks like, right? So the column represents all the genes, right, that are being taken care of here. And then, the rows represent all the genes that are being taken care of. And then the column represents the various groups. So here we have 2 groups, right, with 5 steps. So we have the COVID-19 infection group, and then the mock, which is, so we have the experiment group and then the control group, right? And each of these groups, we have 5 steps for each of these groups. But normally, most of these data sets, you open it and you see, instead of the genes, you numbers, right? And then these numbers are gene numbers that are being provided from the sequencing tool that you used, right? So to start everything, so let's first set our working directory to where we have our data sets, and then what we first do is to import our RNA Seq data set, right? So we can look at our RNA Seq data set by clicking on this, and then we realize that this is how it looks like. So we have all our genes in the row column, and then our group samples in the row, and then our group samples in the column, right? Now we try to convert this data frame into metrics. So we use this to convert the data frame into metrics, and then we look at the head of the data frame. So it is the same thing that we saw here, but here, instead of it in the form of a data frame, we've now converted into a form of a matrix. So before we are dealing with several groups, right? We looking at this example, let's say if we have this group CC01.20 COVID-1, right? We have similar one here, CC01.20 MOC1, right? So we try to compare these two groups since they were all taken at the same time point, right? But different sample groups. We try to compare them to see if they are correlated or if there's some kind of difference between these two data sets. So we look at the one in 5 there. So CCO2, this one, CCO3, 20 COVID-2, and then CCO3 20 MOC2. So we try to look at these to see if they are really correlated or there's some kind of difference between these two data sets, right? And yes, so that's what we have here. And we realize that if there were no difference between these two data sets, we realize that this would just give us a straight line, right? But we can see some variations here. So that means the genes within these two samples, right, are expressed differently. So they have different transcripts being generated from these genes, right? So now, since these data sets, or you can also even do the same thing for, let's change this to say 1, right? And let's change this to 6. So when you change this to 1 and then this to 6, what you are comparing here is the first COVID infection transcript here, which is this column, the first column here, right? And then its corresponding control data set, or let's say, non-COVID infection data set here. So we are comparing this, the CCO2 20 MOC1 against the CCO120 COVID-1 against the CCO120 MOC1, right? So we can do the same thing for this, and we're gonna see this figure is going to change. Yeah, so you see, so we are comparing the 1 and then against the 6, and it's the same thing. So that means these data sets, they are differentially expressed. So the next thing that we need to do in this process is to assign conditions to the data set, right? So when we look at the data set, we realize that the first 5 columns represent the experiment, right? So these data sets or these samples contains, they are being infected by COVID-19, right? And then the next 5 are mock data sets. That is, they have no infection. So we try to assign these conditions, and then we tell out that our first 5, right? These are the experimental data set. These are the ones with COVID-19, right? And then the next 5 are the ones without COVID-19 infection. So we assign these conditions, right? And then R does that for us using this factor function. So after setting these conditions, now it's time for us to analyze our data set with this DSeq2. But to do that, we need to get these libraries to use. So we have to call on these libraries. So we call these libraries, that is the DSeq2, the R-Color-Bury, the ggplot2 for our plots, the dplyr for other analysis, and then gplot. So now we create a data frame to initiate our DSeq2. So we just run that. And before we do our analysis, we need to create a DSeq data set, right? So we use the DSeq data set from metrics because, we first moved our data frame from data from table form to matrix form, right? So that we can set the conditions and then, plot to see how these samples relate. So now we need to send it back to metrics. So we use this DSeq data set from metrics, right, to create, go back to data frame. Yeah, so now it has converted to data frame. Now we can look at it. And then here we see that our data frame, right, the RNA Seq data set do have 60,639 elements, and these elements correspond to the various genes that have been recorded, right? So we have 60,639 genes, and then there are various transcripts, right? Given various groups, that is the COVID-19 infection and then the non-COVID-19 infection. So we run the DSeq pipeline. So what this DSeq pipeline does is, normally most of these data sets are normalized, right? Because these transcripts are some of them are very huge numbers. And normally you can use these huge numbers using this DSeq pipeline, and then it will help normalize these transcripts for you. But the data set that we are using was normalized using TPM, but still the DSeq pipeline will still normalize them and goes through all the DSeq processes. So here we check number of rows. So the number of rows represent the number of genes that we are considering, right? So as I said earlier on, we have 60,639 genes, and that is being given here when we look at the number of rows. But most of these genes, you realize that most of these genes, you look at their transcripts and then they are zero. So that means they produce no mRNAs, right? And then when we use the, in our analysis, it's gonna, there's gonna be a lot of noise in our data set, which is gonna affect the model. So what we are doing is we are removing all genes that has counts less than 3, right? And then we are discarding them, because if their transcription, or if their transcripts are less than 3, it means they didn't produce any mRNA, right? And this is based on the researcher or the one doing this analysis. You can use 5, you can use 3, depending on how you want to shape your data set or how you want to trim your data set. So here, what I'm doing is, okay, we are trying to get all the genes that has counts greater than 3, right? So we do that, and then we check our rows, and then we realize that now our genes has reduced from 60,000 to 19,000. So that means there are a lot of genes that have transcripts less than 3, and then these genes have been discarded. We are not considering them in our analysis, right? So the next step is to visualize what is going on. Visualization speaks a lot about how data behaves, right? So we are trying to plot these groups, that is the experiment group and then the control group, right? To understand or to see how each of these genes within a certain boundary, how they behave towards each other. So here we use the principal analysis, which is the PCA plot to do this. So we plot and we save it, but because we want to see the plot here, that's why I'm plotting this again. It's the same thing, but I'm using this to save it. So when you run this code, it's gonna have a plot saved in your directory, the directory that you've assigned to it earlier on. So we plot this, and then here we can see, using a PC or just drawing a line right here, we can differentiate between our experiment group, that is the samples with COVID-19 data set, and then the samples without the COVID-19 data set, which is the control group, right? And then realize that the samples with COVID-19 data set, they stack together, right? As compared to those without COVID-19. right? So this says a lot about our data set. So now we can see that, okay, now we can differentiate between genes. If we go through these steps, we can differentiate between genes that are up-regulated or down-regulated between these two groups, which is the control, those without COVID-19 data set, which is the control, and then the experiment, that is the one with COVID-19. So we use we move on, and then we use this the detect groups to look at from all the column names, right? We are trying to get this a function that we're going to use to construct our DSeq to get us the genes that are up regulated and then the genes that are down regulated. So run this. And here, so what we are doing here is to use heat map, right, to visualize the up regulation and then the down regulation of the genes between these two groups. So now what I'm saying here is, okay, so I'm telling the code to bring me or to generate the top genes based on their standard deviation, and then using the Pearson correlation. All right, so yeah. Okay, so let me zoom this. Yes. Sorry. Yeah. So now this heat map presents the various genes, right? And then they are grouped, or they are samples. So we have the mock 5 mocks, and then the 5 COVID. So these are the control, and then these are the experiments, right? And then how these various genes, are expressed at each of these groups, right? So here there was supposed to be let me read it from here. Yeah, so there was supposed to be a color key, but because it's too large, the figures, we can't see the color key here. So this is the color key. So from green to red, where green is downward expression, and then red is upward expression, right? So here we can see that when we look at these virus here, these genes here, the virus ORF1AB, the virus ORF110. So these are genes, and these are the ones that we can really differentiate between their control group and then their control group and then their experimental group. That is the ones with which has been infected by COVID-19, right? Even though others too, this we can't. Looking at this gene here, which is DMBT1, we can differentiate between their expression from this mock group here, and then the experimental group here, right? And also when you look at their mock group, which is the CCO120 mock 1 and then CCO120 mock 2, right? You can differentiate them between the so the CCO1, CCO120 COVID-1, and then CCO120 COVID-2, right? So from this, we can tell, this tells you a lot about some of these genes that you think are being, affected by COVID-19. And then this, we will give you an idea that maybe after the differential gene expression, you expected to see any of these genes as the top gene that is being downward or upward expressed in your data set. So now the next is to get our differential expression results. So this differential expression is just trying to tell us the various genes, right? Which of them are, let's say upward expressed in one group and then downward expressed in another group, but in orderly manner. So we use the results function for that. And so this is running, yeah. So we look at the various genes. So what is the results does is looking at all these genes, right? So it looks at its base mean, its log fold change. So log 2 fold change is trying to look at the change between the experimented group and then the control group. If there is some kind of change between it, based on log, right? And then this is the log 4 change standard error, and then this another statistic that is being used in the DSeq pipeline. So here, what we are trying to do is, because some of these log 4 change are very small, right? We are trying to discard or trying to say that if the log 4 change is all those weights, because when you run the results function, the log 4 change that it uses is 0.001, right? But we are trying to increase the threshold to 0.01 so that we can change the log 4 change. And then those that are folds that are below this, we say that, okay, those are not significant, and then we don't need those. And then those that are greater or equal to this, those are the genes that we are interested in. So we run this. Yeah. Yes, so now we are our log 4 change. We are looking at those with log 4 change, which is greater than 0.01, and realize that out of those 12,000 genes, right, 888 of them do have log 4 change. That is, these 888 of them are upregulated, and then 877 of them are downregulated. We have outliers, which is 47, and then the low counts are 4,888. So here, what we are trying to do is now to plot, to visualize our those genes that are significant in our DSeq pipeline. Let me copy this. So all right. All right, so it's supposed to plot here. Yeah, so here we visualize our genes. So there are these genes here, the blue genes here, those are the top here. These are outliers, and then those genes, the blue, that are between, that are above or below this straight line, the zero line here. So these are the genes that are upregulated and then downregulated in our data set. So here we take data frame based on adjacent p-values. So here, what we are doing is that looking at the p-values that are being generated from our DSeq output, we are saying if the p-value is less than 0.1, right? If it is less than 0.1, then we should name that as significant, right? But if it's greater than 0.1, we should name it as not significant. Right? So here we want to look at genes that are way upregulated and then downregulated in our data set when we output all. So we do that, and Yeah. So, yeah. Projects. All right. Oh. So, yeah. Yeah, so this image is too big, but we can see it here since, yeah. Yeah, so here we can see a vast difference between our not significant genes, our significant genes, and then these genes, there's no difference between the control group and then the experimented group. So these red genes here, so these red genes from this volcano plot shows genes that are not significant based on our adjacent p-value threshold, and then the black genes here shows genes that are significant or that are upregulated in one of the group and then down-regulated in the other. But when you look at these red genes, when you look at groups, that's the control group and then the experimented group, you can't tell whether if they are upregulated or down-regulated. Right. So here we try to sort them and then put them in terms of, okay, so we are looking for the genes with the most differential gene expression between the control group and the experimental group, so in the form of ascending order. So the top genes, that's the ones with, that is really being affected. When you look at these two groups, you can differentiate between this gene when you look at these two groups. So that should come first, and then continues to the last genes that when you look at the difference, these two groups, you can't see any difference in them. So we try and then sort out the highest log4 genes. Right. And then we can get our top gene. So the top gene is so we can get our top gene, right? And then the top gene is the gene that is having the highest difference, or that you can really different, or is you can differentiate it between the control group and then the experimented group. Right. So we can plot our top gene, and then so this will tell us that when you look at the control group, right, these are the normalized count of our top gene, which is the virus N. So this virus N. But when you look at the experimented, when we get these transcripts from COVID-19 infected patients, we realize that the count increased, right? But during the control group, when there's no COVID-19, the transcript or the count of these genes is very low. But when there is a COVID-19 infection, right, the count of these genes is like somewhere more than 1,000. So the same thing, we can also look for other genes. So when we open these RES mutate, so this RES mutate is like a data frame containing all the genes, right, that are differentiated, that you can differentiate between their control group and then the experimented group in ascending order. So the top gene is the first one, which is the virus N, followed by all these virus genes, right? And then we have this MMP9 here. So when you look at, we can look at the last gene that is here. Let's take this gene here, right? Okay. And then we can see if we can differentiate, the plot can differentiate between the control group and then the, put it here. Right. So you see, when we plot for this gene, which is FXB046, you see there's no difference. You can't really differentiate between the control group and then the experimented group. It's like they are all within the same space, right? So it means this gene is not being affected by the COVID-19 virus, right? But when we take the genes that are being, let's say some of these genes here, which is this virus gene, right? And then we look at it, plot count. So yeah, we look at it, plot count. All right. Yeah, so we can see, we can, same as the top gene, we can differentiate between the control group and then the experimented group, right? So after that, we can save all these genes as a file, as a CSV file or as a text file. And then looking at the various genes that have been affected by COVID-19, now you can try to look at, okay, how are these genes, what is how are these genes affected by COVID-19, and then what is their interaction between these genes and then the other genes, the interaction between these genes and other proteins, and this helps in the developmental process of getting drugs. So that is the end of this session, and I am open to questions and suggestions. Thank you.
Thank you, Daniel, for this elaborate presentation. And now we'll be moving on with some of the queries. So I can see that there are already many questions raised up in the chat from our participants. So let's move on to that. The first question that has been asked is, how do we predict or identify the expression data used in R? So would you want to take that question?
How do you predict to identify the expression data used in R?
Yes, so I see that you have directly used the expression data in R. So the question I'm presuming is, how do we obtain this expression data?
Oh, okay. So, sorry.
So I'm sharing my screen, sort of, and this is the pipeline on how do we identify the expression data. Is just to help you put your thoughts together. So if you would want to explain this.
Yeah. So this is the workflow, that is the sequencing workflow that I talk about, right? So you prepare your biological library or library preparation of biological samples, and then you sequence your data, right? So you sequence your data, and then the data, yeah, so you sequencing, it pass through sequencing to produce these reads, right? And these reads are input as a FASTQ file, right? And then the read, you align this read to the reference genome, and then the HiSat tool is also a kind of software that you can use to do this, right? And then after doing this, then after referencing the reads to your genome, then you can try and then look at your data set to see if there's some kind of contamination in your data set, right? By using this, I think there's a software, strength type, right? And then also understanding how the risk looks like, right? And then after that, you can do some normalization of these risks, because most of these risks are very big numbers, and then you normalize your reads, and then you do your differential expression here, and then in now. So this is a workflow, and then most of these are being done using command line, because when you are trying to go through this workflow using R, even there are some R codes, and then there are some R libraries that are being used, but normally there's some kind of touch, touch and then read, there are some kind of online or available tools like this, high site to bowtie, and then others that are being used in getting this RNA-seq workflow done.
Well, thank you. The next question that we have is, how is EDGE-R or any other tools like DEG-seq2 are different than DEG-seq used in this study? Would you want to take up that question?
Yeah, so I think EDGE-R also goes through the same format as DEG-seq2, right? But most people use EDGE-R if their sample size is, I think, is less than 3, right? So 3 samples. So looking at this, we have, say, 2 groups with, say, 5 samples each, right? You can use EDGE-R, you can use DEG-seq2. Sometimes it depends on the scientist or the one, doing the analysis, which of the two he is familiar with. But there's no significant difference between EDGE-R and then DEG-seq2.
Thank you. I hope that answers that question. Another question that we have from one of our students is, in drug discovery, how RNA-seq data is helpful, or I'm presuming that means how could RNA-seq data be implemented ultimately into drug discovery? So would you like to take that question?
Yeah, so we know that there are the same genes in each cell, right? And then these genes transcribe, they are expressed differently in each cells to come up with to generate tissues and then others, right? So when you're able to understand the kind of genes that have been affected by the COVID-19, as we've done here, so from this differential gene expression analysis that we just did, we've realized that the virus N, right, is the top gene that is being differentially expressed within the control group, that is the group without COVID-19 infection, and then the group without COVID-19 expression, and then infection, and then the experimental group, which is the group with COVID-19 expression, right? So now, after looking at this, now we look at the genes, their interaction between themselves, the various genes around them, right? The interaction between these proteins, right? And then based on that, right, drugs can be discovered based on understanding these genes, how they interact between themselves, because we need drugs that can, if COVID-19 infection is trying to increase the expression of these genes, we need genes that is going to repress the expression of these genes, right? So understanding these genes and then knowing how they express between these two groups will help us understand the chemicals that are needed in this drug creation, and drugs that are needed to help express, or help express or repress these genes.
Well, thank you for that answer. I hope that satisfied that question. Before we proceed further, I have this little query that I would like to take up with you. And the question is, the expression data that you have used in this study, do you have any idea like which strain it is, or which country was it from, or because we're talking about SARS-CoV and it created a havoc in around the globe. So I would like to for you to shed some light on which kind of stain it was, and where was it from, and things like that. A little detail on the data set that you took.
Okay, so if I can, let me share my screen. All right, so let's go to this. Yeah. All right. All right, so this is the gene expression omnibus database, right? And this is where the I got the data set from, right? So here, the summary here is gonna tell you the experiment type that they use, the sequencing type, that is the high throughput sequencing, and the summary of the data set, the overall design, the contributors, the platforms, the samples. So we have 10 samples, the COVID-19, CCO 120 COVID-1, and then to CCO 320 MOC1. And this is the RNA Seq data. So this is the count, right? And this is the normalized one. If we really want to understand what goes on, we can just go to this run select, the SRA platform. Right. Yes. And let's click on the first one. Yeah. Yes, so there's the selection, the cDNA, the sources, which transcriptomic strategy RNA seq, and there's a bulk RNA sequencing, not a single cell. Okay, and I don't know if I'm answering your question or.
This is pretty close. Thank you.
Yeah. All right. So the server here will link you to the RNA seq data set, and then from there you understand the summary and then all the stuff they use in generating this data sets.
Okay, Daniel, thank you so much for this nice presentation and all the descriptions and, attending to all our participants' questions.